Welcome back, everybody. I hope you enjoyed the coffee, the biscuits, and also potentially completing that bingo card. A reminder, in order to be able to get one of those wonderful prizes for the bingo networking, you need to have handed it in by 4 o'clock today, please. So don't miss out if you'd like to be part of that. And just one other housekeeping piece for those who are using phones. Would you mind ideally not, if that would be all right, only in that the light is distracting to some others within the audience? We'd appreciate it. Thank you. At ZSL, we are obviously incredibly proud of the work that our fellows do and their contribution to conservation. And we're very excited to bring 4 to the table today for you to hear their voices. And I'd like to introduce first, if I may, Dr. Patrick Lewis. He's a leading neuroscientist at the Royal Veterinary College. Professor Patrick Lewis studies how protein malfunctions lead to devastating diseases in humans and in animals. And his talk sheds light on the mysterious world of prion diseases and what we're learning about both their detection and also their treatment. Dr. Lewis.
Thanks very much. And actually, thanks very much for giving me a chance to talk about this at the meeting. I've really enjoyed the meeting already, and I think there's a lot more to look forward to. As Biff said, I'm going to talk about prion diseases and really marking the fact that we're now 40 years on from the description of bovine spongiform encephalopathy or mad cow disease. As was mentioned, I'm based at the Royal Veterinary College. This is just around the corner, about 15 minutes walk away from here. So it's nice to be able to come up to Regent's Park. But my interest in the prion diseases actually started many, many years ago. And I studied across the road over at UCL at the MRC Prion unit, which really grew out of some of the concerns for public health around BSE. Actually, I spent most of my time working on scrapie, which is a disease of sheep, which I'll talk about in a few slides' time. And I was working on trying to understand the molecular mechanisms of the disease. This is a younger version of me working in the containment laboratory at the MRC Prion unit, fiddling around with some scrapie samples to try and understand how the infectious agent replicates. But the focus of my talk is really trying to giving a historical perspective on BSE and what we have learned from that, but also giving you an insight into some of the frontier science that's going on, looking at prions and trying to understand what they do. And the starting point for this really was the first description of BSE in cows. It occurred back in 1985 and was reported in scientific literature, veterinary literature, in 1986. And what you can see here is what happens with BSE and why BSE was picked up by the farming community and by the veterinary community. BSE is a neurological disease. It impacts on the brain, on the function of the brain, and actually devastates the neuronal networks within that brain. One of the key symptoms of that is the development of ataxia. This is a movement disorder. It impacts on an animal and also on humans' ability to control their limbs. And you can see this. This is a clip from an American news item on BSE back in the 1980s. And you can see here that actually this is an animal that has really lost control of its ability to move and control that movement. Now, not long after the clinical description of BSE, it was realized quite quickly that actually what is happening is that there is spongiform degeneration of the brain. What is happening is that cells in the brain are dying, and they are leaving huge vacuoles, great big gaps in the brain, which leave the brain looking like a sponge. One of the other names for the prion diseases is the transmissible spongiform encephalopathies. And actually, one of the best examples of this that has been known for many, many years is scrapie. I was talking in the break about the fact that scrapie was actually described over 300 years ago. It was reported in Hansard. The sheep industry, of course, has historically been incredibly important for the economy of Britain. And it was discussed in Parliament. that started from the veterinary college, and John McFadden, who was principal of the college, described scrapie and gave a very detailed description of what it looks like and how it occurs back in the very late 19th century. And this is a photo of a sheep with scrapie that John McFadden published in the Journal of Comparative Pathology. As I say, it's been around for a very long time. This was a report in the 18th century, going back to 1750, in Germany, describing a disease of sheep impacting on their ability to move and eventually resulting in their death. As happens with BSE, actually one of the impacts of the neurological decline in the prion diseases is the loss of an ability to swallow. And so sheep, cows, and also humans who develop prion diseases eventually are no longer able to sustain themselves because they're no longer able to eat. It gives you a real insight as to the devastating impact of these disorders. It's called scrapie because of what it does to the sheep. This is a video of a sheep that's developed scrapie. And one of the key symptoms in sheep is that they develop an intense itching sensation, and they are compelled to scrape up against fences, against gates, against bushes. And this is why they develop that sort of emaciated loss of wool that you saw in that photo from John McFadden. Similar to BSE, when you look in the brains of animals with sheep with scrapie, they develop vacuoles. They develop this spongiform degeneration, and they lose actually quite a major mass of their brain tissue. What was realized about 50 or 60 years ago now is that when you look in the brain, you see this spongiform degeneration. What you also see is the accumulation of protein aggregates. This photo over here on the right actually dates back from the 1960s and is a photo of fibrils that have been isolated from the brains of sheep, showing accumulation of amyloid. Some of you may have come across amyloid in the context of Alzheimer's disease. And this aggregation of protein is actually thought to be similar to what occurs in the brains of individuals with Alzheimer's disease. To return to BSE, just to remind you all of the impact of BSE as an explosive epidemic, this is a time course over from 1985 to 2000. The first cases of BSE were recognized in 1985, 1986. And what you're seeing here is a monthly report of the number of cases in the UK. This is data from what was the Ministry of Agriculture, Food and Fisheries, or MAT, showing the number of cases that were reported over the course of months and years during the late 1980s and 1990s. What you can see is that this epidemic exploded. There were thousands of cases being reported every month, and hundreds of thousands of cows were being diagnosed with the disease. But actually the impact of that in terms of farming, and Javier and I were talking a little bit earlier before his talk about the One Health concept and how this fits in with the prion diseases as a case study of this. And actually the impact in terms of agricultural farming was much, much larger. And millions of cattle were killed, they were culled and incinerated to try and stop the spread of the disease. And eventually that did work. Around the early 1990s, at the peak of the epidemic in cows, you can see that it started declining. And this was due to a really aggressive approach to try and eradicate BSE in herds. And as you can see here, actually the cases declined and declined and declined. If you were to see this continued now, and we still monitor, one of my PhD students is involved in monitoring new cases of BSE. There are occasional cases, 1 or 2 cases a year, that still occur, but it's nothing compared to what was seen in the past. And this was due to the eradication of entire herds with the identification of individual cases. I don't have much time to talk about it now, but as you'll be aware, there was a lot of discussion at the time about the potential transmission of BSE to humans. John Gummer was quite strident in saying that British beef is safe. We now know, and there's a very detailed body of evidence supporting this, that BSE could be transmitted under rare circumstances to humans to cause a disease called variant CJD, which mirrors many of the symptoms that are seen in cattle with BSE. Some of the slides I think have not come through, unfortunately. But what I'll move on to now is to give you an update on some of the other things that are happening in prion diseases. Thankfully, BSE is not a thing of the past. There are cases occurring year on year here in the UK and overseas, but they're very rare. And because we monitor for them, actually we're able to control and prevent their spread. This is a deer, and Javier and I again were talking about this as a potential as a case study for One Health. There's actually a similar disease to BSE in deers and cervids called chronic wasting disease. This, we think, was naturally occurring in deer populations in the US, was first described in the Rocky Mountains back in the 1970s. And in contrast to BSE, where you saw that explosion of cases, it was really a rapid burning epidemic. Chronic wasting disease started in the 1970s, but actually over the course of many years, and this is data from the US Geographical Survey, you can see the individual dots highlighting where these cases occur. Over the course of years, it has very, very slowly spread all the way across the continental United States into Canada. And actually, we now know there are cases a little bit closer to home. You can see that actually the number of cases is spreading and spreading. This is a slow burning epidemic spreading throughout the US. In contrast to BSE, as far as we're aware, there are no cases of chronic wasting disease being transmitted to humans. But of course, given the example of BSE, this is something where there is a great deal of concern and actually quite a lot of research going into at the moment. It has come closer to home. There was a little bit of talk about deer populations in Scandinavia. And this is a map showing cases of chronic wasting disease that have been reported in Finland, Sweden, and Norway. So it is a disease that is spreading beyond North America. More recently, actually, we're beginning to recognize prion disease as another mammalian species as well. This is a report from 7 years ago now, reporting the first example of a prion disease in camelids, in dromedary camels in Algeria. And there's a strong possibility that these diseases actually haven't been recognized in the past because people just haven't been looking for them. But this gives you an idea that actually beyond cattle, beyond farmed animals, beyond some wild animals, actually there are examples from other species. And actually the slide which didn't come through gave an example from here at ZSL. There are a number of animals, a number of exotic ungulates, but also of big cats that developed prion diseases during the 1990s upon exposure to BSE. A number of cheetahs up at Whipsnade developed what is termed feline spongiform encephalopathy. So it's something that directly links to ZSL and to the zoos here. I wanted to take one example of an aspect of how the technology has allowed us to understand more about what is causing prion diseases. Prion diseases are almost unique in that they don't have a nucleic acid basis. What they're caused by are those protein aggregates that are found within the brains of individuals who develop prion diseases, of cows, of sheep, and so on and so forth. And where there's been an amazing advance, I did my PhD about 20 years ago now, there's been a huge advance in our ability to look at these aggregates and actually visualize them in 3 dimensions. And the development of a technique called cryo-electron microscopy, basically using an electron microscope but doing it very, very cold, allows you to take those fragments of aggregated protein that I showed you a few slides ago and actually map them at an atomic level. So you can look at individual atoms and see how they relate to one another. And work over in the Rocky Mountains. This is the National Institutes of Health Laboratory led by Byron Coey. And here in London at the MLC Prion unit over at UCL, where I studied for my PhD, has allowed us to actually purify those fragments and aggregates and look at them in 3 dimensions. These haven't come through particularly well, but what you're looking at here is a molecular map, an atomic level map of those fibrils in 3 dimensions. And you can actually zoom in on that and see that at an individual atomic level. And so what you're seeing here is a molecular map of those fibrils, and the shape of those fibrils, the shape of those protein aggregates is what is really driving both different types of prion disease, but also driving their ability to transmit, to pass from animal to animal, and from species to species. There's a huge amount of work going on in this area at the moment. My PhD student working at the Animal and Plant Health Agency is working to try and purify scrapie fibrils from the brains of sheep with scrapie and look at them in 3 dimensions in this way. The hope is that actually we can gain insights not just for scrapie, but also for BSE, for chronic wasting disease. There was a recent report of this published from the Rocky Mountain Laboratories in Colorado, and actually really understand how these diseases are moving from one cell to the next, but also from one animal to another and from crossing the species barrier as well. I'm running out of time, so I'm keeping very, very resolutely to time. I've got a poster next door, so if you're interested in chatting a little bit more and asking any questions, do feel free to come over and chat to me then. I'd just like to thank the Animal and Plant Health Agency. The PhD student I have working on this is based at the Animal and Plant Health Agency, so we work collaboratively to try and understand it. And thank you very much for your attention.
Thank you very much, Dr. Lewis. I do encourage you to go and find out more and also converse with Dr. Lewis further. Our next speaker is Richard Kimblin, KC, who, due to unforeseen circumstances, terribly sorry, is not able to join us in person today, but has pre-recorded a speech for us. Richard Kimblin KC has a foundation in environmental science and a legal career that spans UK's highest courts. He brings with him a deep expertise in planning and environmental law. His talk explores how nature conservation law is applied on the ground through a case study of bats, habitats, and housing developments in Wiltshire.
Now, you might think that this short presentation is about bats.
I'm going to do
Indeed,
that.
I would like it to be.
Can my team at the back turn that up anymore?
I don't know anything about bats. So we're going to have to stick with the buildings. And the topic is, in fact, about the relationship.
I think one moment, I appreciate you can't hear this, it's far too quiet.
And how we find places for people.
Technical support's on its way. I tried that one. It was on max. We had to have one technical blip, didn't we? And we've got to 12:05. We're doing well.
A short presentation is about bats. Indeed, I would like it to be. Unfortunately, I don't know anything about bats.
Back up to there.
So we're going to have to stick with the buildings. And the topic is, in fact, about
talk amongst yourselves about what you learned in the last session. What I could say, actually, while we resolve this one, is that during the lunch break, we will be showing a film of the Darwin frogs and their restoration rescue back from the field to Venezuela, severely endangered by the chytrid fungus.
Ultimately,
am I getting a thumbs up about any volume? Hannah's at the top doing her very best.
Southwest of England. But there's a bat. And what we're interested in is the favourable conservation status of bat species themselves, and also the sites.
I suggest what we do perhaps is go to our next speaker and we'll see whether we can resolve that.
Special areas of conservation.
Right. With apologies to Richard Kimblin, I think we will have to revert to him in due course when we can sort that volume issue.
That's it.
So, Sarah's going to be up quicker than she imagined, so she can take a deep breath. But I would love to welcome Sarah Bradley, who is a veterinary nurse and very passionate conservationist. Sarah's worked with animals such as guide dogs in the UK, all the way to rhinos in South Africa, and has been a fellow for a long time, alongside her father, who's with us here today, whose father himself brought him to the zoo. So a long-standing connection as fellows with ZSL. And in this talk, she's going to share her first-hand experience with Wilderness Foundation Africa and the power of connecting wildlife conservation with community well-being. Sarah, over to you, and your volume control is within your gift.
Good afternoon, everyone. I'm delighted to be here today to be able to talk to you about a conservation charity the Wilderness Foundation, and in particular their Forever Wild Rhino Protection Initiative. I work as a veterinary nurse for Medibet, who support the charity through client donations and fundraisers. It's not mine. The other way. So every year there's the opportunity for Medibet staff to experience firsthand what the conservation work that the Wilderness Foundation do in the Eastern Cape of South Africa. And last year I got to go. So long wait to go. I've been really wanting to do some conservation work since being a veterinary nurse and obviously having great interest in animals. So I travelled to South Africa and I stayed with the project coordinator and past Medibet veterinary surgeon, Dr. Will Folds, at his home in the Amakhala Game Reserve. I'm going to start off by talking a little bit about some statistics, which I actually found quite scary when you actually get there and start learning about the poaching problems out in South Africa. Every day a rhino is killed for its fawn. That's actually down from every 8 hours. So the impact that the Wilderness Foundation is having on poaching in South Africa is actually working at the moment. There's also been a slight trend in numbers. There are 17,500 white rhinos left in the wild in Africa and around 6,500 at the moment. And this has been a slight increase in the last few years. So again, that's having a positive impact on numbers. What was really shocking to me is in the space of 7 years when poaching was at its highest, there was a 9,000% increase in poaching, which is staggering to imagine. These are the current figures to show how what the poaching levels are like in South Africa at the moment. As you can see, there's the peak was around about 2014. The lower figures after that actually represent a drop because of COVID lockdown, and they're starting to sort of rise again at the moment. Since 2010, the Kruger National Park has lost around 8,000 rhinos. Of the 586 rhinos poached in 2023, 499 of them were in South Africa alone. So far this quarter of 2025, 103 rhinos have been lost to poaching, 65 of those from national parks. 15 rhino-related arrests have been made during this time. So rhino horns are just made of keratin, like our fingernails, so it's incredible to believe that a single horn can fetch as much as £200,000 on the black market. So however, the bulk of this money doesn't actually go to the poachers because the horns are shipped out to Asia where they're used for traditional Chinese medicines and jewellery. So the Wilderness Foundation is working to take direct action against poaching and creating suitable habitats to support new populations. One initiative they're involved in is the natal area where there has been a 67% decline in poaching since the dehorning programme has started. They lost 325 rhinos in 2023, so that's a 67% decline since then. So it's having a really positive impact. Doesn't hurt them at all. It looks quite brutal with the chainsaw, but they don't feel anything. As I say, it's like cutting your nails. So next slide is quite graphic. So I don't know if anyone, you might find this, not this one, actually. That's not horrific at all. So this is the mission statement of the Wilderness Foundation, where they are saying that they're doing direct action against anti-poaching, wilderness management, and most importantly, developing young leaders, because conservation at the end of the day has a great foundation in education. So it's the next slide that you might find distressing. So one of the important parts of the Foundation's work is to support law enforcement, and Dr. William Folds often gets called out to give evidence for poaching incidents. In 2012, he was called to a site where 3 rhinos had been brutally poached. And in an effort to remove as much horn as possible, the nasal cavities had been destroyed because they tried to remove the growth plate as well. One particular rhino was found to still be alive. We watched videos of this out there, and I can honestly say there wasn't a dry eye after seeing this. After years of enduring pioneer treatment and operations, Sandy's not only still alive, she has had 5 calves and of her own, and is even a grandmother. And despite her horn never growing back, she has become a beacon of hope in the fight against poaching. Most poaching actually happens with the cycle of the moon, because they will take advantage of the full moon to have the natural light. Usually it's a group of 3 people, 1 to shoot, 1 to hack the horn, and 1 to run if they get caught. Usually the one who runs has the least convictions, because unfortunately, if they do get caught, they often get let out on bail and reoffend again straight away. They usually only have 1 shot as well to remain undetected. So this is why rhinos sometimes aren't actually just killed with one shot. So how is Medivet helping with the fight against poaching? So we collect donations from clients, and we've been doing it for the last 10 years. And so far, we have raised over £1 million in the first 5 years, and this has helped to go towards anti-poaching tracker dogs, treating and raising over 50 orphan rhinos and poaching survivors. They also provide flea, tick and worm treatment to local community dogs and cats, neutering over 100 dogs, providing over 2,000 rabies vaccines, and also contributing towards the upkeep of anti-poaching planes. So I'm going to talk a little bit now about what I actually got to do, which is the exciting bit. So after a long journey, we finally arrived and got to go out on our first game drive straight away. And we were rewarded after about 20 minutes of seeing fantastic rhinos in this picture. So this is a female and her calf. There was a male following at a short distance, because when the calves are around 3 years old, the rhino will usually mate again. Every rhino is microchipped and has a tracker fitted to its front leg. They all have notches cut in their ears so that the rangers can identify them. We also saw, I've got some pictures later on, we also saw some lovely cheetahs, 3 brothers who were participating in a bit of grooming each other, which was just lovely to watch. So Amicala Game Reserve is 18,000 acres in size and has the big 5, which are leopard, lion, elephant, rhino and buffalo. And you never quite know what you're going to get to see at all. On our second day, we went out, we got called out to a neighbouring reserve to treat a rhino that had had a fight with another rhino and had some gouge injuries from its horns. So it's very different from what I'm used to with cats and dogs. Obviously the size of them to start with. So before any procedures, we all have to have a meeting to discuss drug doses and make sure that we carry the antidote with us, because 1 drop will kill a human. So we all have to have really good safety briefings before we go out. Once the rhino has been darted, which is usually by helicopter or by buggy, we have to get to them within about 8 minutes. It takes about 4 minutes for the drugs to start working, and we usually find them out in the open, and as soon as you dart them, they're off. And I didn't realise rhinos could actually run so fast. And you lose them really quickly as well. So you often have to get the drones up to go and find them. And you've literally got about 8 minutes from darting to find them, because we have to administer some drugs to stop oxygen levels falling too low before we get to them. So this is me with the pulse oximeter. So once we find them, we have to put a blindfold and earplugs in first off, so that they don't get worried about any noises. Their eyesight isn't that great, but obviously if they come round, it's better for them not to be able to realise what's going on. So they're put on, and then we establish an IV line. Being out in the field, we don't have all the bells and whistles that we have in clinic, and so we're very basic monitoring. We generally look at the colour of the blood to make sure that they're oxygenated properly, and obviously have the reading on there. Due to their size, you have to turn them quite often as well, so that they don't get tissue necrosis because they're so heavy. So the great thing about the trip is that it's not just open to the vets and nurses at Medivet, anyone can go. Everyone just gets stuck in and does their job. This is Lee from recruitment, who is about to get well stuck in. Don't think I need to tell you what he's about to do. But joking aside, it's really important that every animal that we deal with, we have, we collect samples. We take blood, feces and tick samples, and this helps us to build a comprehensive database for health reasons, and obviously any breeding programs that we might go on ahead with. Day 3. We were changing a tracking device on a rhino. Unfortunately, the dart went in at an angle and he didn't get the full dose. So he ran a long way before we found him. So we didn't actually get to him till 11 minutes. So it was a really worrying time. We were all in awe of Will, who just leapt out of the vehicle, ran over and gave a intravenous injection to a half anaesthetized rhino. So it was very impressive, but to him it's all in a day's work. But for us, who've never seen this sort of thing before, it's just amazing to be that close to them. So this one was having a tracker refitted to his leg because the batteries had gone, and as they grow we need to put a bigger size tracker on them as well. It just looks like a bracelet. And we also microchip the horn, so if he does get poached and it's picked up at security in any of the airports, we'll be able to see where that horn actually came from. We spent a lot of time in the classroom learning about poaching and what the foundation is doing to help. Day 4, we were out in the community. It's really important that we have good relationships with the locals. We were providing vaccinations against rabies and neutering as well. And I can honestly say, operating on a table that half an hour later was having lunch served off was a first for me. I don't think I'm ever going to have to operate in someone's kitchen ever again, hopefully. And then day 5, we had to go and retrieve 2 cheetahs who decided that they were going to eat out in another reserve. They'd done this several times before. Unfortunately, 1 in 5 cheetahs die under anaesthetic, so it's really vital that they don't get moved too often. Absolutely incredible day. I think this was my favourite. Just being so close to the cheetahs was amazing. We also visited the Tanglewood Conservation Area, which is a biodiverse conservation corridor project where they are linking up areas of barren land between reserves to help with corridor flow for breeding programs. This was the hardest day, I think. We were having to do some tests on some buffalo. Nothing ever happens quickly, so it takes hours to prepare for all of this. We were up at 5 AM, had to drive 2 hours to do this. These buffalo were being transported to a new area, so had to be tested for TB. And again, we took all of the samples beforehand. Later that day, we got to go up in the plane, which the anti-poaching plane, which Medivet contribute towards, to see how fencing is maintained and how they can see where the animals are from the sky. These are just a few of the pictures that I took. I mean, I could stand here for hours showing you the pictures we took. Just part of the amazing trip with the cats. On our last night, we had fantastic barbecue at South African Braai, and the locals danced and sang for us. So it really was a once in a lifetime experience. That one you can see on the rhino, that's his tracking device on his leg there. And that's Will over at the side there, and a view from the plane. Last day, we went to see the sunrise before departing, which was just a very magical moment. I felt like I needed a lion cub to hold up on the top of the cliff for that one. And we were joined by the Helping Rhinos CEO, Simon Jones, then that day as well. So it was interesting to see from another charity's perspective and how they're all working together. So that's the end of my talk. And just a little reminder that no one needs a rhino horn except a rhino. And I've just put some information up there for some websites if anyone's interested in getting some further information. Thank you.
Thank you, Sarah. What a fascinating talk about you taking your veterinary expertise out into some less clinical environments, clearly, but essential to helping the rhinos. Thank you. Our next speaker is Dr. Himani Siriparpu, who has 2 decades of experience across continents. Dr. Himani combines both scientific research with also community-led conservation. And her presentation today is going to highlight the efforts to save India's endangered bird, the white-napped tit, through habitat restoration and sustainable practices in the Aravalli Hills. Dr. Himani.
Good afternoon, everyone. And I'm going to present my work, environmental awareness and habitat restoration. But before starting, I would like to tell you background of this study. Why I chose this environmental awareness as conservation tool is before starting this study, I did kind of gap analysis and why we need specific this tool to conserve this bird species. And in a country like India, where local communities are highly dependent on forest resources, where if I need to conserve this species, I need to first take consideration of local communities because they are hugely dependent on natural resources from the forest. And this particular species is highly endemic to is thorn forest. Thorn forest and is a kind of indicator species of thorn forest. And this bird is facing extinction, not only from anthropogenic pressure from the local communities, but from the natural nesting competition from other species. Earlier, it was people said that, or references says that nesting competition is just because of a particular species is Brahmini starling. It's like common starling, this type of bird, and it's a whole nester bird. So whenever a woodpecker makes a hole in a tree, it was supposed to breed, but because of the nesting competition, Brahmini starling takes the hole, and this bird species is not able to breed. So that was the thought from the previous studies. But before that, I did another study where I found out that this species is not facing breeding, nesting stress from other bird species, but from a tree mouse and wasp species and kind of squirrel species. They are occupying the natural holes, and this particular bird species is not able to breed. And because of its declining population, this bird is categorized in vulnerable from IUCN, and it's highly protected species in Indian Wildlife Protection Act 1972. It's very specific to tropical thorn forest, and previously it was supposed to say that habitat degradation, fragmentation is the major cause for its population decline. And second significant factor was population decline was nesting from particular bird species. And I said earlier also, it's not only one bird species, but from other mammalian fauna like squirrel and tree mouse and some wasp, and all these species are taking up the nest boxes. And for this, to find out this competition from other species, I did earlier studies. It was like nest box. I put up artificial nest boxes to find out the actual reason of declining population. My study area is in Aravalli Hills. If you see, if you can see, this starts from further north from Delhi, and it goes down and until here, Gujarat. It's one of the oldest and one of the longest hill range in India. And you can see the major part is covered in Rajasthan. This western part is completely desert. It's known as Thar Desert, and this part is like forest, and it has different type of forest, like dense forests and open grasslands, thorn forest, and even some kind of moist deciduous forest also. This is the forest type where this species reside. So to this is from my previous study where I tried to put up some nest boxes, and this project is funded by the Small Grant Foundation UK. And I tried to find out the natural competitor for the bird species. So, and this bird species is called as pike-tit also because of its white and black color. And for this particular studies, I did mapping of thorn forests through ArcGIS. Then I did the assessment of anthropogenic disturbance in terms of collection of natural resources from the forest, like fuel, wood, and fodder, because local communities highly dependent on natural resources. In terms of fuel wood, they are going and cutting deep in the forest and bringing this fuel wood outside, and grassland, because most of the communities in around the areas are like they have livestock in enormous amount. They just let them free in the forest areas and they go and cut. And for this, it's an experimental study where I try to develop a model fuel and fodder plot outside the study area with the permission of local authorities and village communities. And I did this as a restoration plan. I did this plantation activities and creating awareness among local communities, different stakeholders, and different group of people. So can you see? That's how. And this data collection was done like anthropogenic pressure. I did this actually whole study was in and around the study area is like 6 villages. I have chosen 6 villages for this particular studies because they are highly dependent of this particular forest. So you can see the way they are carrying out. And it's not that for their particular daily need they want it. For they are stockpiling these type of wood logs because there is a tourism area around the this habitat. And if I don't know, but after China's Great Wall of China, there is another fort there which has 32 kilometers wall around that area, and people come from around the world to see that particular wall. And it's wildlife resort are all around the area. So these local communities not only taking these resources out for their own consumption, but to sell them to the local areas, local resort managers. So if you see earlier, this habitat fragmentation is shrinking. Its habitat is shrinking and getting fragmented. So earlier, this community used to walk like 3 to 4 kilometers, but the habitat is fragmented so much. Now they are walking like 9 to 10 kilometers in the forest to collect all this fuel wood. And this, for this fodder, they just leave their cattle and these livestock free in the forest. And a country like India, where communities are dependent on the forest and they have lots of rights on the forest, you can't just stop them by not taking the forest resources from their local area. So my intention was to talk to them, and I can't reduce, I can't ask them, don't go to the forest and don't cut the fuel wood or firewood. I just can ask them to divert a little pressure from their forest and go to their own area, because every community and every village, they have allocated land which government provide them, and it's called as gocha land, where they can develop their own resources. But because it's easily available natural forest, so they go and just cut and let their cattle free. So for that, my thing was to discuss with local communities in different level. Like there's a women group where I used to discuss, and then some different male. And sometimes it's going to the school and communities and talking to them about their particular species, because this is an indicator species. And if I protect that particular indicator species, I can protect this thorn forest. So you can see, and it's a different focus group. And if you see, there's only men and there's only women, because the community I'm working with, they don't sit together in a public gathering. So I need to do some meetings in the nighttime, sometime in the daytime, is because some socioeconomic changes in their So before these things, when I came to know that I can divert and I can aware them about this proper conservation thing, I talked to them about this particular species, why you need to protect it. And when you're working with local communities, taking their confidence is a big deal. And I talked to them and I convinced them, why don't we develop their own wasteland and their gotcha land? And we will provide you full support, like providing seeds, providing other water support to irrigation and everything. They were ready. And I tried to do this experimental study in 6 of their plots. And for that, we did a selection of site. This is, can you see, this is completely wasteland. This is completely wasteland, and it was encroached by invasive species which grow very frequently and very easily, and they don't use it. This particular species, if you see, they don't use it for fuel wood or firewood. It's called Prosopis juliflora, and it just spread. You just cut it and it spread easily. So my thing was clear this land, clear these invasive species, whatever present in that area, and then bring the native grass seeds. See, with the help of local people, local youth, this is the village head. With local head, we got the native grass species, and we try to do all this irrigation and she's sowing in their land. My purpose is to show them the potential of their own grassland where they can grow their own resources instead of going deep into the forest. I know it's just a little bit, but at least showing them the potential is necessary and getting them engaged in these activities. So this local community and these young people, they were volunteer with me and they're still carrying out this work. So this is the plantation activity as part of restoration I did. It's in their land and this is in a school. So you can see these grasses. And what my suggestion was like, they can let their cattle open and go and graze. But once cattle go into the thing, cows graze, they just cut from the top. But when goats and sheep, they go into the field, they just browse from the root. And if they keep on going because of their hoof, the soil gets eroded and you can't, another seed can't grow. So my thing was like, why don't you do rotational grazing? You do in one plot, you cut the grass, go to the another plot and then let it grow and generate a seed bank. So that was my thing here. And you can see, and somehow they were managed and they were convinced that this is the best practice and they don't need to go 8 to 9 kilometers to get the wood. And the plantation I did, this small trees, still they are not that big, so that can go. But at least they know that if we conserve this species, we are conserving our thorn forest habitat, which is just in the vicinity of their habitat, their area. So this, you can sharing of resources, like you can see here. They're sharing the resources, means all the communities and all the village people, they know this is my plot, I need to share, and they share among themselves. Today my cattle will go, or I'll cut in the grass like this. So they are sharing. And earlier it was not like that. They were like, no, first I will go, because everybody has sense like, it's my plot, I'm free to go. So now at least they have a committee among themselves who will go, which part of the area will go and collect the grass. So they are collecting and they are following this rotational grazing practice until now. So see, all these studies were discussed with local state forest department also and different stakeholders. And if you see all this effort, because the study was divided in 2 parts. One is to stop natural competition for the bird. It was done through like providing the nest boxes, artificial nest boxes in their territory. And second was doing this model plot development. So local authorities and local forest department was involved, and they included all this conservation plan in their own plan, and they're following it still now. So all this recommendation has gone to forest department, and they're still carrying out these studies. And all this is well sustained and had ensured the long-term survival of vulnerable white-necked tit and its habitat. It's just taking community into consideration, getting them involved into this all the practice, and involve forest department, all the stakeholders sit together and discuss about why we need to save. And this model is kind of successful. People are still following. It's almost 10 years now, and still people and my volunteer is still conserving that area, those grasslands. And that's how this species is still surviving this area. And I got to know that they can see the bird is breeding in artificial nest boxes too. Thank you for listening and your questions. I think I'm too quick. If you have any question or something, you can ask me freely. Thank you.